Hi, so this is the part B of the chronic inflammation lecture. And what we're going to talk about in the second part of this lecture is ultimately this is specific processes that are occurring during the process of chronic inflammation. And we'll talk about what's happening within the tissues and specifically what's happening in the tissues within the context of periodontitis. And this is a, excuse me, this is a really good example of a disease where you get destruction of not only the soft tissues, so things like the gingival tissue, but also the destruction of the hard tissues as well. So basically the underlying alveolar bone of the tooth. So we'll talk about the processes that occur and specifically the role of these MMPs, so matrix metalloproteinases, and also the role of osteoclasts and osteoblasts in bone degradation. So what happens in periodontitis, which as we should know by now is a chronic inflammatory disease, basically you see changes not only in the soft tissue, so basically destruction of the gingival tissue when you shift from oral health into oral disease and specifically periodontitis, but you also see destruction of the hard tissue as well. So these are just some x-rays showing that you get recession of the alveolar bone, basically arising from destruction of the bone itself. So this diagram here highlights the processes that occur during the process of periodontitis and the onset and development of the disease. So the first 3 points here, which are highlighted up here, are things that we've talked about in great detail in previous sessions, where you get initiation of the immune response, which is driven by recognition of the microbes and the components of the microbes, so basically antigen recognition. You get the effector responses within the immune cells, which can lead to recruitment of other immune cells, so neutrophils and macrophages play an important role here. And then finally, you get activation of the immune cells. So that can be either innate immune cells or adaptive immune cells. But what we're going to talk about in the second part of this session is basically what happens downstream of that in the context of tissue destruction. So these three processes can arise. So we have Rank-L production. So we'll talk about Rank-L in a few slides. But basically the activation of osteoclasts and the reduced function of osteoblasts. So basically the balance between the function of osteoclasts and osteoblasts is important to consider when you're talking about bone remodeling and bone resorption within, in this example, the alveolar bone. So this is basically what's happening when the hard tissue is being destroyed within periodontitis. Within the context of the soft tissue, the MMPs, so these matrix metalloproteinases, have a really important function, a really important role in driving remodeling of the soft tissue. And we'll cover these points in a bit more detail in the coming slides. So in the next few slides, we're going to talk specifically about tissue destruction, soft tissue destruction. So basically destruction of the gingival tissue that is associated with periodontal disease. So these MMPs, I don't think I've touched upon them before in any of my sessions, so it's a bit of a new concept for you. These MMPs are produced by many different types of immune cells. So things like fibroblasts, dendritic cells, lymphocytes such as B cells, T cells, neutrophils, macrophages, and also endothelial cells that line the blood vessels. What you need to be aware of is that all of these different immune cells can produce different types of MMPs. And these MMPs, there's many, many different types. You don't need to learn the function of all of them, but their overarching function is described in this next slide. And their main function is to basically remodel the extracellular matrix. So the extracellular matrix that is found within the connective tissue that underlines the epithelial surface. So extracellular matrix is predominantly made of protein fibers, mainly collagen. It's produced by things like fibroblasts, which are found within the connective tissue. And the primary function of the ECM is it's a complex structure that supports the cells, and it basically supports the immune cells that are resident within the tissue underlying the epithelium. So remodeling of the extracellular matrix by these MMPs is important not only in cell migration, so this idea of promoting migration of cells from the blood vessels into the tissue in certain infections. So it allows immune cells to actually move through the tissue and enter the site of infection. Also, they play an important role in this process of angiogenesis. So angiogenesis is ultimately the formation of new blood vessels within a tissue that's repairing. And MMPs play an important role in this as they basically make room within the tissue for the new blood vessels to form. So moving on to the hard tissue destruction, and specifically in the context of periodontal disease, the destruction of the alveolar bone that is underlying the tooth, that keeps the tooth in place. And it's this idea of bone remodeling and the balance between bone formation and bone resorption that you need to be aware of. So bone formation, the technical scientific name for that is osteoblastogenesis. And this is driven, as you could probably tell by the name, by osteoblasts. Bone resorption, on the other hand, is driven by osteoclasts, and the technical name for this is osteoclastogenesis. So what you need to be aware of is that bone is constantly remodeled during adult life, and you actually get a new skeleton forming every 10 years from this process. And generally speaking, the formation and resorption of bone is maintained in sort of equilibrium. So there's no net loss of bone in a healthy individual. And this diagram highlights that quite nicely. So you have osteoclast recruitment and activation. So these osteoclasts will resorb the bone, degrade the bone. Osteoclasts will then be removed, and osteoblasts will then function. And osteoblasts will basically remodel the bone and form new bone in the place of the bone resorption. And then eventually you'll get a new bone being formed, and this process is constantly occurring. There's this cycle of bone resorption versus bone formation within health. Of course, in certain diseases such as periodontitis, this process is not in equilibrium, and therefore you get more bone resorption than you get bone formation. So if we go into a bit of detail on the two different types. So firstly, we'll talk about bone resorption, which is ultimately the degradation of bone. It's mediated by osteoclasts, and you need to be aware that osteoclasts ultimately differentiate from macrophages. And what I'm going to introduce to you in the next few slides are the components, the three components that are really important in this process that are produced by either the osteoclasts or the osteoblasts. So the first component is this protein, this ligand, this rank ligand that is produced by osteoblasts. So this rank ligand, although it's produced by osteoblasts, will actually activate a receptor known as rank, which is found on osteoclasts. Okay, so osteoblasts will produce this rank ligand. This rank ligand will then bind to the receptor that's found on osteoclasts. So basically the osteoblasts and the osteoclasts will communicate with one another during in a healthy situation in a certain scenario where the process of bone resorption and bone formation is in equilibrium. And so this process is quite carefully controlled within tissue homeostasis. And this production is controlled, and thereby you do not lead to any excessive amounts of bone resorption. And of course, as I've said, this balance, this homeostasis can be tipped within a disease such as periodontitis. So we've talked about rank L and we've talked about rank. The third protein that I want to talk about is OPG. So OPG is also produced by osteoblasts, okay? And this OPG is really important in actually trying to control the function of RANKL. So OPG will inhibit RANKL function, therefore and thereby controlling bone resorption. And this diagram here quite nicely highlights that. So these are the three components that we've just talked about. So we've got RANKL. So this is a protein that's produced by osteoblasts. We have the RANK receptor, which is found on the osteoclast. And then OPG is a protein that's secreted by osteoblasts that basically inhibits and prevents the RANKL from binding to the specific receptor on the osteoclast. And it's ultimately the balance between OPG and RANKL, which is really important in maintaining this tissue homeostasis, maintaining this balance between resorption and bone formation. And as I've said, in health, this is a balanced process. And the ratio between RANKL and OPG is important in modulating this process. However, in certain diseases, such as periodontitis, you do start to see a shift in this ratio, which basically results in a tipping of the balance between bone loss, or bone resorption, and bone formation. And this is highlighted quite nicely in these two figures here. So this figure on the right-hand side shows that in tissue homeostasis, you have a nice balance between RANKL and OPG, and basically old bone is resorbed and a new bone is formed in its place by the osteoblasts. In disease such as gingivitis, you get a shift in the ratio. And this shift in the ratio can actually tip the balance so that osteoclasts are functioning more prominently than osteoblasts, and therefore you get more resorption than you get formation, which can of course then lead to recession of the bone, and in this case recession of the alveolar bone. And this figure here is from a scientific paper from a few years ago now, and this highlights quite nicely how important this ratio is. So in healthy individuals, the ratio, there's a specific ratio where you have quite a low level of RANKL and quite a high level of OPG. In gingivitis, so acute inflammation that can resolve itself. Gingivitis, again, you may have a slightly higher level of RANKL and also a lower level of OPG, but generally speaking, the ratio between the two is still quite low. And that's important to consider because, of course, in gingivitis, you don't get any resorption of bone. However, in chronic periodontitis, shown here on the right-hand side, you start to see a shift in the ratio, where basically you have more RANKL being produced, less OPG being produced, and thereby the ratio between the two is a lot higher. You've got more RANKL being produced by the osteoblasts, and we know now that RANKL will basically drive this process of bone resorption by activating the osteoclasts. And that is just an idea that you need to be aware of, is that the balance between RANKL and OPG that is produced by osteoblasts is important in controlling osteoclast function. And the final thing I just want to highlight here is that what happens within the osteoblasts to produce this RANKL? Well, basically you get stimulation of RANKL production by certain microorganisms such as P. gingivalis. So this is a specific mechanism alongside all of the other mechanisms that P. gingivalis has developed to basically drive the process of bone resorption in this case. And you've seen this diagram before, and I've just changed the labels on it just to highlight my point here, is that P. gingivalis can actually be recognized by osteoblasts via this receptor recognition, which we've talked about in great detail before. Downstream signaling cascades will happen that lead to RANKL production. RANKL is a protein. It's essentially an effector response by the osteoblast in response to recognizing P. gingivalis. Excessive amounts of RANKL being produced will basically imbalance the ratio between RANKL and OPG in a very similar manner to as I've shown here. You're getting more RANKL being produced in chronic periodontitis. And that is in part due to the way that P. gingivalis can modulate this process. And that's just something to be aware of here. So quite a brief part B of chronic inflammation. Hopefully it made sense. Just a final recap on the session. So there's different classes of chronic inflammation. These can either be due to the formation of granulomas, such as OFG, or not, such as periodontitis. M1 and M2 macrophages are important in the process of chronic inflammation in balancing this process of tissue injury versus tissue repair, and that's something we'll revisit in the next session. In periodontitis, tissue destruction of soft and hard tissues can occur. When you're talking about soft tissue destruction, this is your MMPs, so your matrix metalloproteinases that can remodel the connective tissue that is underlying the actual gingival epithelium. And then on the other hand, you have destruction of the hard tissue as well, this process of bone remodeling, basically the equilibrium in bone remodeling being disrupted within the disease. So I've introduced to you the idea of osteoclastogenesis and also osteoblastogenesis. So osteoclasts can resorb bone, osteoblasts can form bone. And the balance between RANKL and OPG, so these are two proteins that are produced by osteoblasts. The balance between these two proteins is essential in the idea of alveolar bone destruction, where basically if you have more RANKL being produced and less OPG being produced by the osteoblasts, then you get more bone resorption and eventually more alveolar bone recession, destruction and recession. So as always, if anyone has any questions, please don't hesitate to get in touch with me. But that concludes the 8th session entitled Chronic Inflammation. In the next session, what we'll do is talk about tissue. We'll talk about tissue healing and repair in the 9th session next week. Thank you for your attention, and I will see you next time.